Welcome back. In this video, we're going to discuss the 4 levels of protein structure. So we were talking in the last video how amino acids bond together through peptide bonds, and then they form a linear structure, a linear order of amino acids. For a protein to be in its final form, it forms a much more complicated structure where it folds, and it folds in such a way, there are rules, it folds in such a way to sort of get the amino acids that like to interact with each other closer together. There are also other proteins that help proteins fold, so it's sort of like the catcher in the chicken and the egg situation, where sort of which came first, the chaperone proteins, which help proteins fold, or proteins themselves. The forces, or rules, that control protein folding are controlled by something called intermolecular forces, or IMFs, which we learned about in chemistry. In this sort of video, we're going to talk about the role of these IMFs in making proteins fold. So there are 4 levels of protein structure. We're going to go through each of them. The tertiary structure is the most important one, but the first one is called primary structure, and we abbreviate that. Pen is not on. We abbreviate that one circle, which means primary structure, and we're going to talk about each one of these individually. Then we have secondary structure, which is abbreviated like that, tertiary structure, which is abbreviated as such, and then quaternary structure, which is abbreviated as this. Each structure, the protein undergoes further sort of manipulations or folding to form its final structure. So as we said, the first level of structure is known as primary structure, and that is simply the linear order of amino acids, the order in which they're bonded together. And they're bonded together by peptide bonds. As it takes forever to write. So essentially, if you had a string, if each box was an amino acid, the order in which the amino acids are bonded together is the primary structure. This may be lysine, which is a positively charged amino acid. This could be proline. This could be glycine. These are all different amino acids. And so forth. And the order in which they're bonded together is their linear structure. They usually often refer to it as sort of like beads on a string. The order in which the amino acids are bonded together, but it's important that it's bonded together by peptide bonds. The next level of protein structure is secondary structure, or as we abbreviate it to 0. I'm going to define secondary structure and then sort of use a picture to explain it. It's a little bit more complicated than primary structure. So secondary structure is caused by interactions between atoms not in the R group. And usually these atoms are called the backbone of the protein. And the interaction is actually a specific interaction. The specific interaction is hydrogen bonding.
We will be meeting in the 4th floor presentation room at...
Hope you enjoyed Mr. Goldblum's interruption there. So the specific interaction that causes secondary structure or intermolecular force is hydrogen bonding. And if you remember, hydrogen bonding is between a hydrogen and a very electronegative atom, so either F, O, or N. Remember, we showed it with lines, but it's a interaction between a hydrogen and then a very electronegative atom. And because this is so electronegative, and this is not as electronegative, this is a partial positive charge, and this is a partial negative charge, and because of that, they attract each other. But secondary structure is due to this hydrogen bonding, okay, between atoms that are not in R groups. And we're going to take a look at this in a picture to hopefully make more sense. So if we take a look here, this is a sort of a structure of a protein with many amino acids bonded together. You can sort of see, here's the R groups here. There are groups over here. Notice that the interactions is a long chain of amino acids bonded together by peptide bonds. There are interactions that occur along the backbone. Here, the C double bond O is hydrogen bonding to the NH of another amino acid. That's maybe on another part of the molecule. Same thing here. We sort of have a C double bond O, hydrogen bonding to an NH of another amino acid, separate amino acids, maybe somewhere else on the chain. And they form these interactions, and as a result, the protein folds, or forms a different shape. There are 2 sort of shapes, or main shapes, that a protein forms in secondary structure. One is known as the beta-pleated sheet, which is here, and the other one known as the alpha helix, which is here. Secondary structure, hydrogen bonding between backbone atoms, not R groups, okay, causes the formation of either a beta-pleated sheet, depending upon which amino acids are there. We're not going to get into the details of that. And also the alpha helix. So just to summarize, we have secondary structure, which forms either the alpha helix or the beta sheet, and it's caused by interactions between non-R group atoms, also known as backbone atoms, and they form hydrogen bonds. This amino acid here is hydrogen bonding with this amino acid here, but it's in a different spot of the long peptide chain, causing it to fold. Next, we're going to talk about the most complicated form of structure, which is tertiary structure. The third level of structure, which we have, is tertiary structure. Tertiary structure is by far the most important structure in terms of like complexity and sort of how the protein flows overall. In general, if we were going to define tertiary structure is caused by intermolecular forces, IMFs, between R group atoms, or group atoms in the R group. We're going to sort of take a look and switch to a small animation to sort of explain tertiary structure. So we sort of see here, this here is the linear structure of amino acids, or its primary structure. The line represents lots and lots of amino acids bonded together. If we press and magnify, you can see along the line there are these peptide bonds that are holding holding is not the right word, bonding the amino acids together. If we click further, you'll see what happens is we start seeing the formation of the alpha helix and the beta sheet. And if remember, these are due to hydrogen atoms between atoms that are not in the R group. So between the C-double bond O and the NH of various amino acids. Now what's going to happen in tertiary structure, okay, is that these alpha helixes and beta sheets are going to fold closer to each other. That's in the most stable way. The R groups on these amino acids are going to interact with the R groups on these amino acids to form the most stable structure. And we'll sort of see that happen. So we sort of see here there are R groups here that can interact with the R groups here on the amino acids and to form the most stable structure that exists. So if we go back here, we're going to list sort of the intermolecular forces that are involved. So this is tertiary structure is caused by IMS between R groups that cause the protein to fold further. That cause the protein to fold further. One of the most important interactions or forces that's involved in this are called ionic interactions. And what happens in an ionic interaction is that the negatively charged amino acids, or the acidic, so negatively charged or acidic ones, fold next to the positive or the basic amino acids. So in an ionic interaction, the acidic amino acids, the ones with the acidic R group, fold next to the positive or basic amino acids, and that sort of neutralizes the charge. That makes the protein more stable. Next, we have hydrogen bonding, like we had in the secondary structure. And hydrogen bonding is it occurs between polar amino acids. So two polar amino acids will hydrogen bond with each other. They have oxygens and hydrogens that can hydrogen bond. Then we have one of the most important interactions. It's called the hydrophobic effect. So in the hydrophobic effect, the non-polar amino acids, they don't like water. And proteins are in your body, so they're surrounded by water. So what happens is the non-polar amino acids fold inward, away from water. So if you have, what ends up happening is sort of all the non-polar amino acids fold inside so they can sort of surround each other and protect themselves from water, so that the least number of hydrophobic amino acids are in contact with water. Kind of what you see with oil when it separates from water, while all the oil is settling on the top, only some of the oil is in contact with water. Same thing with the hydrophobic effect. All the non-polar amino acids go in the middle to be protected by the polar amino acids from water. The last interaction we have, which is a very specific interaction, is called a disulfide bridge. occurs between cysteine amino acids. And I'm going to go back to the table with the amino acids. Just give me a second. So if we look at this amino acid here, this is going back to one of your earlier slides. This is a cysteine amino acid. And what can happen with a cysteine amino acid is that if there are two cysteines, let's say like there's a one cysteine all the way over here in the chain, it can fold next to the cysteine here, and the two sulfurs can bond to each other and they lose the hydrogens. We'll see this in an image on the next slide. So if you see here, I'm flipping back and forth between slides. Here we have sort of one chain and the chain continues. And here you'll see this is a cysteine residue. It has the cysteine R group. And here are what's going to happen is it's going to lose those hydrogens, and then these sulfurs will find a disulfide bridge. Two sulfurs bridged together by a bond. The picture down here, if you look at this, is the linear sequence of amino acids, or its primary structure, and the yellow dots represent cysteine amino acids. If you see what happens, the protein folds so that the cysteine amino acids, their R groups, can bond to each other in the fashion here, and causing the protein to fold and form a shape. Sort of the details here, it sometimes forms intermediate shapes while it's folding, and then eventually forms the final shape. But the point is that these cysteine amino acids, they like to fold next to each other, so they can form a disulfide bridge, which releases energy when it's formed and makes the protein more stable. So, just to summarize, tertiary folding, or tertiary structure, is caused by interactions between R group atoms, due to certain intermolecular forces, which cause sort of the alpha helices and the beta sheets of the secondary structure to fold closer to each other. Just so we go back to the animation that we were playing before, here we're at secondary structure. If we fold it one more time, you're going to see that this alpha helix is going to fold on top of the beta sheet. And the reason why that is probably the residues, which is sometimes what amino acids are called, here interact positively, or there's a good interaction between the residues on this part of the protein and this part of the protein. Perhaps if we will play it. Perhaps here, there is an acidic amino acid here and a basic amino acid here, and they interact with each other positively. They form an ionic interaction, which releases energy, causing the protein to be more stable. So the last structure, which again we've at this point where it's got easier, is quaternary structure. And quaternary structure it's caused by the marker back interactions between R groups of multiple polypeptide chains, and the interactions that happen in tertiary structure are the same as the ones that happen in quaternary structure. So you still have the same interactions as tertiary structure. You have the ionic bonds, the disulfide bridges, the hydrophobic effect, and the hydrogen bonds. If we go back to our animation, you'll see all that changes. So I've actually already pressed it forward, just to sort of talk through it. If we look here, we'll go through again. We have our primary structure, linear structure of amino acids. Here we have our secondary structure, further folding tertiary structure. In quaternary structure, what happens is another polypeptide chain, as these are all connected, all the amino acids, another polypeptide chain comes, and quaternary structure is the interactions, or how these two chains interact with each other, is what quaternary structure is, if you add another polypeptide chain to the structure. I'm going to talk about this tomorrow in class, but one of the proteins that's important, we'll talk a little about tomorrow in class, is hemoglobin. And hemoglobin has 4 subunits, and tomorrow in class we're going to talk about a little bit about the protein hemoglobin. It carries oxygen to body tissues, and sort of how its structure helps it form its job. But you should make sure that you answer the guided questions for this lecture, and I will see you guys tomorrow.